Alright, so in this video I wanted to talk a little bit about the so-called lab leak hypothesis for the origin of the COVID-19. So lab leak hypothesis for the origin of COVID-19. And so this isn't something I had thought about that much. I've kind of ignored sort of the back and forth about whether it came from a lab or whether it came from bats or maybe an intermediary pangolin. Um, but since it's recently come back into the news, I decided to look into it. Uh, and I'll admit right from the outset that I went into the literature thinking I was going to find that the lab leak hypothesis was somewhat overblown. But I was surprised to find that, well, the evidence for the lab leak hypothesis is actually pretty solid. I'm not saying it's a knockout. It's still a hypothesis, and more information has to be obtained in order to confirm it. But I would say that just based on what I've read in the scientific literature, which is what I'm going to be covering here, I'm not going to go too much into sort of the politics of it. I just want to look at the science and what the science actually says. And the science actually shows that this lab leak hypothesis isn't that unfounded, and in fact, I would say it probably has the uh, has the highest probability of being true just based on what I have read. And so the science behind the lab leak hypothesis is based on essentially two lines of evidence. And they both have to do with the spike protein, which is what the coronavirus, what the SARS-CoV-2 uses to enter human cells. And so the first line of evidence is the unique furin cleavage site, sometimes also called the polybasic furin cleavage site, or the polybasic site, or the multibasic site, or oftentimes FCS for short. Those all mean the same thing. And so the furin cleavage site has a 4 amino acid insertion. So there's a 4 amino acid insertion. And so, so what is usually said about this, or what I don't know if it's usually said, but what I've found said about this are things like, for instance, that this particular furin cleavage site with this particular 4 amino acid insertion shows up only in artificial conditions. So it is found in influenza viruses, but only in artificial conditions, such as labs and on farms, where there's essentially a large, a large sort of, if you have a chicken here, it passes to the next chicken because they're in close quarters, and you have a high turnover rate of your chicken stock, and so the viruses, it essentially sort of simulates a gain of function experiment in a way, and that it's the only lineage B coronavirus to have one of these furin cleavage sites. And so the second line of evidence is that, I guess this isn't unique. I think I copied it over from my number 2. So it just has this furin cleavage site. It's not unique. It's a known type of site. But the unique, so it's a unique unique is it's a unique sequence in the receptor binding domain. So receptor binding domain, you will often see it abbreviated this way, and this has a affinity high affinity for the human ACE2 receptor. And so this is also said to be the other sort of primary bit of evidence for the lab leak hypothesis. And so just to sort of get a bearing here on what I'm actually talking about, so the spike protein is used to gain entrance into the cell by the viral particle. And so in this illustration here, what we have is we have the spike protein, which is this right here. And so this at the top up here is the membrane for the virus up here, and down here is the membrane for the human cell. And so this is showing so the time axis is here. So we have the viral S spike protein here, the spike protein, and it comes and it's in this, it shows this down position right here. So this arm right here has to sort of extend outward so that it goes into what's called the up position, and that allows it to bind onto this receptor here, which would be the ACE2 receptor. And then there is actually a cleavage where all of these S1 domains are cleaved off, and you end up with just this S2 domain here, which then extends outward and sort of grabs onto the human cell membrane, and then sort of bends inward here and allows fusion. So fusion of the cell membrane. So the viral particle has all its contents in here, and so when you get fusion of these membranes, the contents can then go over into the human cell and start infecting it. So with the SARS-CoV-2, it actually inserts RNA into here, and then the human ribosomes and so forth start translating the RNA and replicating it and things like that. Well, it translates the RNA into proteins. Then the viral proteins will actually replicate the RNA. But anyway, so this here shows, this is a figure, so the SARS-CoV-1, you could even put a 1 here, of the 2002-2003 fame, binds to the ACE2. SARS-CoV-2 also binds to ACE2 here, but it can also bind to these other things here, including this furin, as I was talking about, can actually cleave it. The SARS-CoV-1 is not cleaved by furin, but the Middle Eastern Respiratory Syndrome, MERS, is cleaved by furin and this TMPRSS2, this DPP1. All right, and so if we want to get into the evidence for the lab leak hypothesis, and so I guess I should say, what is the lab leak hypothesis exactly? So the lab leak hypothesis is essentially that the Wuhan Institute of Virology is where COVID-19 came from. So the COVID-19 virus was produced at the Wuhan Institute of Virology, or WIV. And so the Wuhan Institute of Virology, which was founded in 1956, was upgraded to biosafety level 4 between biosafety level 4 between 2003 and 2014. And one of the things they study here are these coronaviruses. So they study coronaviruses. And one of the things that they do to study coronaviruses is what's called a gain of function. So gain of function experiment, gain of function. And so this is sort of a broad term for several different kinds of experiments. So probably the two most common ways that this will happen is, so if you have a virus, and we'll call it V1. So one of the ways you can do it is just insert using recombinant DNA methods, insert a sequence, or perhaps you could use site-directed mutagenesis, and then you end up with the virus with some increased gain of function, which is why it's called a gain of function. So you would insert a sequence or do site-directed mutagenesis that would confer onto the virus some new function, and that new function in these type of experiments is often increased pathogenicity, And the other way that you can do this is you can take your virus V1 here. So maybe this is some natural virus. And so what you do is you will infect either cell culture, so cell culture, or some in vivo experiment, so some animal. So you'll infect some animal, and so then what you do is you take these cell cultures where you had the most pathogenicity increase, and then you take that and sort of put it into another cell culture, or you do the same thing with the animal. You take it from the animal, you take the viruses that have the most increased pathogenicity, then reinfect other animals with it. And you keep doing this over and over, what are called passages, as you will see in the literature. And then over some number of iterations of this, you end up with some, we'll call this one V3 because it could be different than this V2 here, which now has increased pathogenicity. And so that's what I was talking about earlier when I said that farms can act like, because it can sort of simulate doing this experiment, because it's sort of like doing this part right here, where on a farm you have lots of animals and lots of animal turnover, and so you can end up having viruses mutating and evolving quite rapidly in a farm sort of environment. And so this is sort of the two ways that you can do gain of function experiments. I don't want to go too much into sort of the policy issues behind this, whether doing gain of function is good or not. So proponents will say, though, that this allows us to find diseases before they happen. So the idea is that you can confer this increased pathogenicity before it actually happens in nature. And then we could actually have data about this before it becomes a problem from a natural source. But of course, the con or the opponents to this say that that we are creating disease that may have never existed in the first place, may never exist. And so then that if these pathogens would have never existed naturally, then we're creating them, and just by creating them, we are introducing a probability that one of them will escape, that some employee at the lab will become infected and then go out into the world and infect other people. And so those are kind of the main arguments for and against this. I would probably land more on the con side, but like I said, I don't want to go into this too deeply. All right, so I'm going to look at, I mean, there's a lot of literature going back and forth about the lab leak hypothesis, but I'm going to look at just a few papers here. So this one is from October 22 of 2020. It's by Rosanna Segreto and Yuri Diagin. I don't know if I'm pronouncing those names correctly, but this lead author, whose other papers I'm going to go through as well, has been sort of, and I think she is, Department of Microbiology, University of Innsbruck, Austria. So she is from Austria. But anyway, she has written quite a bit about not dismissing the lab leak hypothesis. All right, and so I've gone through and highlighted a few things I wanted to mention here, and there will be links to all these in the description down below. So I would urge you to read through these yourself. So here they're talking about bat CoVs have been thoroughly studied and genetically manipulated, and they talk about in order to evaluate emergence of potential novel CoVs, which is doing that gain-of-function research. Researchers have created a number of chimeric CoVs consisting of bat CoV backbones, normally unable to infect human cells, whose spike proteins were replaced by those from CoVs compatible with human ACE2. So they are taking the genomes of CoVs that cannot infect humans and making it so they can infect humans by binding to that ACE2 receptor. These chimeras were meant to simulate recombination events that might occur in nature. And so, as I said, what they are saying is that this type of research has been going on. So we know that there is gain-of-function experiments going on with coronaviruses, specifically to bind to the ACE2 receptors, which is what SARS-CoV-2 does. So such gain-of-function experiments have raised a number of biosafety concerns, which I've already sort of talked about. So in recent years, the field of coronavirology has been focused on pan-CoV therapies and vaccines. So synthetically generating diverse panels of potential pre-emergent CoVs was declared a goal of active grants for the EcoHealth Alliance, which funded some of the research at the Wuhan Institute of Virology in collaboration with laboratories in the USA and other international partners. And so they're saying, this is saying that the Wuhan Institute of Virology is in fact doing these gain-of-function experiments on coronaviruses. All right, so creating chimeric CoVs with novel receptor binding domains has gone on for decades. So researchers have been generating chimeric CoVs for over 2 decades, long before the advent of modern sequencing or genetic engineering techniques. And once again, like I said, I urge you to read through this yourself. I'm not going to read the whole thing off here. Links in the description. Probably the largest reported number of novel chimeric viruses was described in a 2017 paper from the Shi group at the Wuhan Institute of Virology. So they're saying that the largest number of these novel coronaviruses generated using gain-of-function experiments was done at the Wuhan Institute of Virology. So going up here, SARS-CoV-2 shares its receptor binding domain with the pangolin CoV. So although its overall genome similarity is lower to SARS-CoV-2 than to the RATG13, which, by the way, is the genome for the bat coronavirus that is often compared to as being the closest. I think it has like 96% similarity. And the RA is because it's from the Rhinolophus affinis, which is a species of bat. I forget what the TG is, but 13 is because it's a sample from 2013. But anyway, is lower to the SARS-CoV-2 than that of RATG13. The pangolin strain isolated from pangolins has an almost identical receptor binding domain to that of SARS-CoV-2. Down here, recombination events between the receptor binding domain of CoV from pangolins and a RATG13-like backbone could have produced the SARS-CoV-2 as chimeric strain. So it's saying that it could have happened. It's possible that it could have happened in nature. For such recombination to occur naturally, the two viruses must have infected the same cell in the same organism simultaneously, a rather improbable event considering the low population density of pangolins and the scarce presence of CoVs in the natural population. So this natural recombination is something that is known to happen in viruses, and so how that can happen is, I know this is kind of difficult to see, but so one of the ways is called template switching. So template switching. And so what happens is we have here our polymerase, which is polymerizing, is generating this new strain, and this is the template strain down here in the magenta. And this polymerase with its nascent strain can actually detach from the template, and then one of a few different things could happen. What could happen is we could have a homologous template at the same position, and so essentially that it just binds onto another template, or it could bind onto the same template again, but at a position that is homologous to, or the same, it would have like some of the same bases on the nascent strand is what would match to the template. So it has to be a homologous sequence, and then it can keep replicating. It can keep extending its nascent strand. But now, if, for instance, this homologous template, or it is possible you could have a non-homologous one, will be added onto this nascent strand. And so it's essentially like if you have a page with some writing on it, it would be essentially like going on your word processor, clicking right here, and then adding other stuff onto it. So it would go here, and then you have this. You start writing right here, and now you are adding something different afterwards onto your strand. And so that is what this template switching does, and it can allow these chimeras where you have so this page here would be kind of like a chimera page where the first part of it is this black right here. So whatever writing was in the black, and then the next part of it is different, and it has this writing that is in blue here. And so it's the same thing with your sequence. So you have a sequence that was going to here, and then you have the template switching that happened right here, and now the rest of your sequence is some different sequence. And so this is how you can end up introducing new sequences into your virus. And so that is how these chimeras can come about naturally. But this is recombination events. So for such a recombination to occur naturally, the two viruses must have infected the same cell in the same organism simultaneously. And the reason for that is you would need this template. So this template you could say is maybe from virus 1, and then you have this other template here which is from virus 2, and so that is how you would have your virus 1 sequence right here, and then your virus 2 sequence right here. So that is how you get a chimera of those two different viruses. And so it's saying that it's improbable that you would have both the RATG13 like backbone and the pangolin sequence here infecting the same cell. So here, all right, so now we're at the fear and cleavage site, the key difference between SARS-CoV-2 and its closest relative, RATG13. All right, so we have here, yeah, okay. So spike protein of SARS-CoV-2 of a cleavage site activated by a host cell enzyme furin, previously not identified in other beta-CoVs of lineage B and similar to that of Middle East respiratory syndrome, MERS coronavirus. And so I think there's, well, this is the receptor binding domain, not the, actually I have that down here. So yeah, this is the furin sequence right here. So this is SARS-CoV-2 right here, and this RRAR has been introduced. The arrow here indicating where it is cleaved, where you see that the SARS-CoV-1 does not have that, so this has been inserted into it. But then we have the MERS-CoV, and so this does have an R here and an R here, which is more similar to this furin cleavage site than the SARS-CoV. But in one of the papers I read, it showed that these R's here, the more parsimonious way that those would have been introduced is through point mutations rather than this insertion event that happened in SARS-CoV-2. And so even though they look the same, they're not exactly the same. All right, so the ubiquitous expression of furin in different organs and tissues have conferred to SARS-CoV-2 the ability to infect organs usually invulnerable to other CoVs, leading to systemic infection in the body. So it's just saying that because it now it has this furin site and can now be cleaved by the at the furin site by furin enzymes, which are expressed on the surface of cells in multiple organs in humans. That's how the SARS-CoV-2 can infect multiple organs. So the ACE2 is in the lung cells, and that's why the lungs are the way that it usually gets into the human body, but it can spread to other organs. So confirm that the polybasic furin site is essential for SARS-CoV-2's ability to infect human cells. So this is saying that that furin site, the introduction of that furin site is what made it so that SARS-CoV-2 could infect human cells. All right, and the polybasic furin site in SARS-CoV-2 was created by a 12 nucleotide insert. So it's four amino acids, but there are three nucleotides for every amino acids. Each codon is 3, so it's 12 nucleotides for 4 amino acids, coding the PRRA amino acid sequence at the S1-S2 junction. So that PRRA is this right here. So the PRRA right there is what has been inserted into it. Interestingly, the 2 joint arginines are encoded by the CGG codons. So this is another one of those big things of evidence that are often talked about because these are rare for these viruses. Only 5% of arginines are coded by CGG. And the reason for that is, so we can look here, so arginine is coded for by these nucleotides right here, these codons right here, and also these ones right here. So there are 6 ways to code for arginine, and it's saying that this CGG one right here is very rare. And so, interestingly, the CGG and SARS-CoV okay, so the 2 joint arginines, which are rare for these viruses, only 5% of arginines are coded by the CGG in SARS-CoV-2 or in the RATG13, and CGG in the new insert is the only doubled instance of this codon in SARS-CoV-2. The CGG insert includes an FAU1 restriction site, of which there are 6 instances in SARS-CoV-2. So anyway, what it's saying, they have this up here. So it's showing that this FAU1 restriction site is right at this insert here, and it's the authors are essentially arguing that that the serendipitous location of the FAU1 site could allow using restriction fragment length polymorphism techniques for cloning or screening for mutations, as the new furin site is prone to deletions in vitro. So they're saying that putting this FAU1 site at the introduced furin cleavage site is a sign that this is something that scientists would have introduced because then they could use it to test for the presence and absence of that furin site while doing experiments. All right, so therefore the SARS-CoV-2 remains unique among its beta-CoV relatives, not only due to the polybasic furin site at the S1-S2 junction, but also due to the 4 amino acid insert PRRA that had created it. The insertion causes a split in the original codon for serine, which is TCA, in the pangolin and the bat, in pangolin or bat, to give part of the new codon for serine TCT, so part of the amino acid. So what that's saying is if you have writing, like say you have writing that just says like the cat, and what it's doing is essentially you split it right here, and so you end up with the and then CA, and then you have some new inserted thing here, and then you have a T, but what you end up with is you have a CA over here and a T right here in the middle of what you just inserted. So you still have the cat and you still have cat here. And so it's saying that splitting the sequence, it's saying that splitting the sequence, it split the original codon for serine, so it's not exactly like my the cat example, but because it is changing from one of the serine codons to a different serine codon, but it's maintaining that serine even though this codon is being split. And so part of the amino acid GCA SARS-CoV, the insertion of the furin cleavage site in SARS-CoV-2 is not in frame with the rest of the sequence when compared with the pangolin and the bat sequences. Therefore, it is possible to exclude that such insertion could have originated by polymerase slippage or by releasing and repriming. And so polymerase slippage and releasing and repriming is essentially what some of what I was talking about down here. So polymerase slippage would be like if it's going and it's hitting each of these nucleotides, but then it sort of skips over some nucleotides. It sort of moves along the sequence without actually replicating these ones right here. And so it could slip along, or it could actually like come all the way off and then actually rejoin that sequence at some later part. And so this is saying that therefore it is possible to exclude that such insertion could have originated by polymerase slippage or by releasing and repriming, because insertion mutations generated by these mechanisms have been postulated to maintain the reading frame of the viral sequence, which is essentially saying that that it would not be splitting this serine codon because the reading frame of the DNA, so like I said, the codons are 3 bases, so you would have, something like this, and this right here would be a codon, but then the next 3 in the sequence would be another codon, the next 3 would be another codon. And you can see that you could change the reading frame. If you got rid of this one, then suddenly these 3 become a codon, and then these 3 become a codon. And so this is saying that these insertion mutations generated by the mechanisms of the polymerase slippage and the releasing and repriming is postulated to maintain the reading frame of the viral sequence. And so it's saying that that you will not get this change in reading frame like this. If it's going to skip over, so if it's going to skip over a base, then it will end up skipping over an entire codon, and so you are not changing the reading frame on there. So the rest of this paper is a critique of this widely cited paper by Christian Andersen called The Proximal Origin of SARS-CoV-2. So before I even get into this, it is important to point out that Christian Andersen, so this is an archived tweet that he made. It's kind of a famous tweet where he goes through the evidence that we've been talking about here and sort of trying to debunk the lab leak hypothesis. But what ended up happening is, and the reason that I have to look at this on the Wayback Machine is because Christian Andersen he tweeted that the COVID lab leak hypothesis is unlikely, but as pointed out earlier, he had highlighted the possibility of engineered virus in a private email with Dr. Fauci. So those Fauci emails, which I think were obtained through a Freedom of Information request, a FOIA request, show that he had actually been a proponent early on in private emails with Dr. Fauci about the lab leak hypothesis. And when that came out, he ended up deleting his entire Twitter page. And so that's why this has to be found on the Wayback Machine. And so even just already from that, his that calls into question his paper, The Proximal Origin of SARS-CoV-2, and the rest of this paper. And I think this came out before those Fauci leaks, and it goes through and talks about what Christian Andersen has wrong. And so I'll just read a couple of the parts here. So the creation of chimeric viruses has been carried out over the years with the purpose of studying the potential pathogenicity of bat CoVs for humans. In this context, SARS-CoV-2 could have been synthesized by combining a backbone similar to the RATG13 with the receptor binding domain of CoV similar to the one recently isolated from pangolins, because the latter is characterized by higher affinity with the human ACE2 receptor. Such research could have aimed to identify pangolins as possible intermediate hosts. So that's saying that, I mean, when I first started, I thought, are they accusing them of doing it to cover it up? But I think what they mean is that the gain of function research was an attempt to actually find out if pangolins could be an intermediate host for the bat CoV, potentially pathogenic in humans. Subsequent serial cell or animal passage, as I talked about, where you keep reinfecting either cells or animals, is described by Sirotkin, and Sirotkin could have provided the perfect adaptation of the receptor binding domain to the human ACE2 receptor. Then up here, Anderson et al. Also state, based on the work of Elmazan et al., that the genetic data irrefutably show that SARS-CoV-2 is not derived from any previously used virus backbone. In the last 6 years before the outbreak of SARS-CoV-2, the number of potential bat backbones, so backbones meaning like the actual DNA sequence into which the furin site would be introduced. So the backbone is sort of the thing that you have, and then you're applying the modifications to the backbone. So it's sort of the underlying or substrate of whatever you're doing to it. The number of potential bat backbones has been undeniably increased by several bat CoV screenings. Last but not least, bringing RATG13 to scientific attention in January 2020. Other possible backbones could as well still wait for publication. And so that is this paper. Just to be thorough, there is this other paper that is trying to refute this first paper. It's called "There is no evidence of SARS-CoV-2 laboratory origin response to Segreto and Dagan. Anyway, but that's this paper that they're talking about. But then the authors from the original paper put out another paper here where they respond to this response paper and sort of go through and talk about so false premise. The RATG13 was not suggested as a direct progenitor of SARS-CoV-2, which the other paper suggested that their original paper had said. So a weak point of criticism: a bat origin of SARS-CoV-2 with direct spillover to humans is highly unlikely. Tishkovsky and Panshin's analysis of restriction sites in and around SARS-CoV-2's furin cleavage site is flawed. And once again, this will be linked to in the description down below. And so the out-of-frame insertion of furin cleavage site is not proven to be natural by the papers cited in this other paper here. Then another paper, which is actually by Rosanna Segreto and Yuri Daigan, as well as other authors here, goes through some more evidence. So this figure here is just talking about possible paths from, so the wild bat, the natural zoonotic infection, or it could be a live sample taken from a bat and a lab worker got infected. So in that instance, there would be no, there wouldn't be any changing of the genome. It would just be that somebody working with one of these bats in the lab got infected by the bat. So the second one, which is the one that has been most often talked about as the natural origin, is a wild bat infected a pangolin as an intermediate host. And then in the pangolin, the virus was able to evolve in such a way that then it was able to infect a human. And so down here, this C goes through sort of the lab leak hypothesis using the gain of function method. All right, so I'll go through a few highlighted things in this paper. So 3 of the 4 patients. All right, so this is addressing the so the earliest detected cases of COVID-19 were located in Wuhan, China. The Huanan seafood market in Wuhan had at first been posited as a possible location of initial zoonotic transfer from wild animals to humans. However, 3 of the 4 patients with the earliest recorded onset of COVID-19 symptoms had no association with the seafood market, and the ancestral T8782 and C28144 genotype. So that's thymine and cytosine at these positions. Genotype was not associated with the seafood market, and so it's saying that the earliest version of COVID-19 was not associated with the seafood market. And so phyloepidemiological analysis of early cases also discounted this theory. And so they're saying that the idea that it came from the Huanan seafood market in Wuhan is called into question. All right, and so although 2 pangolin coronaviruses exhibited strong binding to human ACE2, binding to pangolin ACE2 was approximately 10-fold weaker in binding to the Rhinophilus pheromequinum, so that's just a bat. ACE2 was very weak with similar relative binding relationships exhibited by SARS-CoV-2. This indicates that neither pangolin coronavirus had adapted well to pangolins and that more research is required to validate the viability of coronaviruses to spread naturally between pangolins. And so it's saying that the coronavirus, so if we wanted to go through this B here, it was not actually that infectious to the pangolins, and pangolins are kind of rare in China because they're actually killed for their weird scaly sort of flesh for sort of Chinese traditional medicines and so forth. All right, and so unlike SARS-CoV in its early and middle phases, so the Chinese SARS molecular consortium, or the evolution of Middle East respiratory syndrome related coronavirus, SARS-CoV-2 exhibits limited diversity across its genome. And so they're saying that the SARS-CoV-2 virus is actually very similar. So there's very little diversity in the genome of the sort of early and middle phase SARS-CoV-2, which says that it came from sort of a single origin, which supports the idea that it would have come from a laboratory. So here, while there is no record of a process of early evolutionary adaptation, SARS-CoV-2's receptor binding domain appears to be highly optimized for binding human ACE2. And so that's essentially just saying that that the SARS-CoV-2 spike protein is highly selective for binding to the human ACE2, which is something that would happen if it was something made in a laboratory. Because in a laboratory, whether you're doing passages or even just using using recombinant DNA techniques, you could sort of focus in and have it be something that really wants to bind to the human ACE2. So substitution of any of the 8 SARS-CoV receptor binding domain residues proximal to the human ACE2 binding interface with the residues found in the RATG13 receptor binding domain were shown to be detrimental to the human ACE2 binding. And so this is talking about the 8 residues that are needed, or that are sort of involved in the COVID-2 spike protein binding to ACE2. And this paper, I think, actually, yeah, this is the interface and sort of the highlighted area here of where the SARS-CoV-2 binds to the human ACE2, and I actually have that right here, so we can look at this. So what I have here in the sort of salmon color down here, that is the structure of the SARS-CoV-2 spike protein. In yellow up here is the human ACE2 receptor. And so the human ACE2 receptor, the structure of it was found with what we have here in purple, the receptor binding domain of SARS-CoV-2 bound to it. And then you can see overlaid over top of that structural alignment with it in salmon is that part of the receptor binding domain from the spike protein here. And then I have up here in cyan the residues that are involved in binding with the ACE2 receptor up here. Interestingly, this one that I have in green, this aspartate here, is, so in this paper it says that substituting any of these 8 residues proximal to the human ACE2 binding interface with the residues were shown to be detrimental to human ACE2 binding. And so what's interesting, it says that these are detrimental, but in fact one of those, so right here, this N501Y is actually this British variant. So the variant of SARS-CoV-2 that sort of came up a couple months ago. It came out of England, and so one of those residues that the paper says, if it's substituted, disrupts the binding with ACE2, has actually been naturally substituted, N501Y here, so that one of those 8 residues has actually been substituted. So that, I don't know if it works against this right here, what they're saying right here, because it might be a very specific substitution that actually did work, and they're just saying that most substitutions would not work. And there were other substitutions in the British variant here that could have perhaps rescued the spike protein from this particular substitution right here. But anyway, we can move on. So, however, tested bat species are poorly infected by SARS-CoV-2, and they are therefore unlikely to be a direct source of human infection. So in addition, in silico, which means computer modeling, just in silico means on a computer modeling of the binding affinity for 37 bat species showed that 8 species exhibited very low binding affinity of their of their ACE2 with the spike protein, and the other 29 exhibited low binding ability. Although the host of RATG13 was not modeled by the group, these results are perplexing as it indicates a significant and unexplained evolutionary distance between SARS-CoV-2 and bats. So this was done with modeling, and I think that was this paper here. So once again, this will be linked to in the description, and this is the paper that they were talking about, I believe. No, this is the Demas one. Forget where. But anyway, they did computer simulations. Actually, I think I have a figure from the paper on here. And so, yeah, this figure here. So this this energy of binding here, and so more negative means it's more has a tighter binding affinity. So we can see that that the best one is to humans. The next best one is to the pangolin here. And then we have to go down all the way to here in order to see the bat. And so it's their modeling shows less binding affinity to bats. However, I did find this paper here where they actually ran an actual assay rather than just a computer simulation. And so in part E here, so multiplex binding assay to measure affinity of different SARS-CoV receptor binding domains to human ACE2. And so what we see is that this pangolin one actually, the pangolin ACE2, so here in the sort of orange, actually binds best to the SARS-CoV-2 spike protein, and the human is actually only second best here. And then the original SARS is fairly close to that right here. So there's a little bit of discrepancy there between this modeling and this actual assay here. And you can see here this is showing the interface between the SARS-CoV-2 receptor binding domain, excuse me, and the ACE2 receptor here, the human ACE2 receptor here. And this right here is a multiple sequence alignment. So we have the SARS-CoV-2, the SARS-CoV, and the RATG13 here. And we see there's quite a bit of sequence similarity, and in the black here is even sequence identity here. But anyway, getting back to the paper. So yeah, this was talking about how the those simulations were showing that there is a low binding affinity to the bats, which is saying that the bats could not come directly from the bats, so they're ruling out this path A up here. Yeah, so interestingly, there is no evidence of recombination events in studies of SARS-CoV-2 by Richard et al. Or by Bobet. This is in contrast with MERS-CoV. Furthermore, there is also no indication of recombination between the subgenus sarbecovirus and beta coronavirus subgena or species of alpha, gamma, and delta coronavirus. Indeed, the subgenera beta coronavirus gene exchange is restricted to members of the same subgroup. The hypothesis of the receptor binding domain of the SARS-CoV-2 spike protein arose via a recent recombination with pangolin-hosted coronavirus. Coronavirus is not likely in poor taxon sampling and is discussed by Wenzel, although earlier recombination and mutations have been proposed. given the sarbecoviruses have not been shown to recombine with other coronavirus genera. So it's essentially saying that the type of coronavirus, the beta coronavirus, um, would not recombine. So if you remember, recombining was this process down here with other types of coronaviruses. And so they're saying that it's unlikely that it could have recombined like that with a pangolin coronavirus. All right, and then talking about, now we're moving on to talking about the furin cleavage site. Indeed, no coronavirus with a spike protein sequence homology of greater than 40% to SARS-CoV-2 has the furin cleavage site. So it's saying that that only other coronaviruses that are very dissimilar to SARS-CoV-2 have the furin cleavage site, and so it's uncommon for anything resembling SARS-CoV-2 to have that furin cleavage site. So the presence of an arginine at the third position, P3, before the furin cleavage site increases the efficiency of the furin cleavage site tenfold. Its presence is also rare, occurring in only 5 out of 132 known furin cleavage sites. So it's saying that that extra the extra arginine here coming before it is not that common. We can actually see here this that in the OC43 and in the HKU1, which are both coronaviruses that cause the common cold, we do have that there, but they would have less than 40% sequence identity with SARS-CoV-2. They just happen to also have these furin cleavage sites. But like I said, they are more distantly related to SARS-CoV-2, so they would be unlikely to be progenitors of SARS-CoV-2. So the proline residue at position P5, fifth residue prior to the FCS. So they're talking about, so it's kind of hard to see, but there's a P right here that is for the proline. So it's 5 spaces before this arrow here, and that's what they're talking about in this part. So the proline residue at position P5 is rare. It only appears in 5 out of 132 known furin cleavage sites. Proline has a restricted phi angular range, so proline is like a weird amino acid that kind of alters the shape of proteins, is what it's saying, which imposes conformal restraints on the peptide chain and results in the separation of the cleavage site from other structural elements. And so if we actually look back at this, so I have the furin cleavage site. We can look at that down here in magenta. And what you see is this is actually separated. There would actually be a loop going between here where that furin cleavage site is. And these disordered regions, these disordered loops are fairly common for areas on a protein that get cleaved because it sort of takes them further away from all this jumbled mess over here so that another protein can gain access to it and then cleave it. In this case, it would be the furin enzyme would come and cleave that so that you could end up getting the this head part of it, this S1, to detach from the S2. And so what this is saying is that this results in a separation. So it's saying that it sort of moves this further out from all this jumbled mess here, which imposes conformal restraints in the peptide chain cleavage site from other structures, facilitating exposure to host proteases. The host proteases in this instance being things like the furin enzyme. And so it's saying that the introduction of a proline sort of makes it even easier for a furin to come in and actually cleave this, which they are saying is indicative of possible human intervention to make it easier for furin to cleave that site right there. And then so if we move over here, so moreover, the furin cleavage site in SARS-CoV-2 is coded by rare codons. That's that GCC arginines they're talking about, leaving it out of frame with the rest of the sequence, thus violating the rules of copy choice recombination mechanisms that populate in-frame insertions. We kind of talked about that from the previous paper. I'm not going to get too much into this. This video is already getting kind of long. So, like I said, these papers will be linked to in the description down below. But essentially, the take-home message here. So we have this lab leak hypothesis, and it's saying that in the spike protein, we have this furin cleavage site with this 4-amino acid insertion and this unique sequence in the receptor binding domain, which gives it a high affinity for the ACE2 receptor. So these are sort of the overall two points of evidence for the lab leak hypothesis. And what I've been talking about throughout this video are these papers that go through the evidence well, the evidence essentially is saying that this furin cleavage site with the 4-amino acid insertion is something that is unlikely to happen in the wild, but is something that could easily be done through gain-of-function experiments. And then the same thing with this unique sequence in the receptor binding domain, conferring high affinity to ACE2 receptor, whereas it has low affinity for the BAT ACE2. So anyway, I've been talking long enough. My voice box is starting to get tired. But anyway, I hope you found this video useful. I hope you don't take it to mean that the lab leak hypothesis is a knockout blow, that it's somehow it's proved, because it is not proved. Like I said at the beginning, I think the evidence is weighted in favor of the lab leak hypothesis just based on my reading of the literature, and especially now that Christian, that scientist Christian Andersen has been sort of shown to be a liar, I guess is the blunt way to put it. And it was his paper, this paper right here, that was kind of a favorite one to cite amongst people who were skeptical of the lab leak hypothesis. So like I said, none of what I said here, none of the papers linked down below prove one way or the other. So nothing is proved. But like I said, I think it shows that the lab leak hypothesis is definitely looking kind of likely. So I'm saying that it's likely. A zoonotic or natural origin is still possible, and I await any further scientific evidence as well as investigations into the Wuhan Institute of Virology and things like that. But anyway, I hope whatever you believed before watching this and whatever you now believe after watching this, you found this video helpful, and I urge you to look at the literature I have linked to in the description down below. I do have papers that are more in favor of the lab leak hypothesis and others that are more in favor of a zoonotic origin for it. But I think from reading both sides that these two things have not been adequately explained by the zoonotic approach, and it's more parsimonious or more likely uh, that these, or it's easier to explain these two things using the lab leak hypothesis. But anyway, I hope you found this useful, and yeah, good luck trying to get to the truth of this.